Will investigate the role of vocal individuality presence in southern female yellow-cheeked crested gibbons in Jahoo, Cambodia. As a background, this study utilizes PAM, or passive acoustic monitoring, using ARUs, or autonomous recording units. PAM is great for non-invasive monitoring of populations, but gaps remain in distinguishing individuals as the data study is in the absence of human observers, thus makes it difficult to study individuality and get data labels if recorded in absence for years. Our target species, southern yellow-cheeked crested gibbons, are endangered. Our study area is Jahoo in the Mondulkiri province in Cambodia. In many other gibbon species studied, there is evidence for vocal individuality. Similar studies on classification accuracy point out distinction in calling variables between paired and unpaired male southern yellow-cheeked crested gibbons. Research will address key questions concerning call detection and classification. Overall, we focus on individual differences. We look at whether the calls to female yellow-cheeked crested gibbons are individually distinct. Ultimately, this gives us insight into territory use, density, population size, and call patterns, which can help with later automated detection. Objectives for this research use supervised classification, test the effectiveness of using deep learning, using automated detection approaches, and determine if we can detect the presence of new females using unsupervised clustering. These comprise of 3 different data sets, one from GPS location, one from automated detection in the middle of territories, and one from acoustic array 3 kilometers away. We can be sure gibbons 3 kilometers away are different as other gibbon group is territorial. It's important that I mention some key definitions of research regarding classification types. In this study, we use both unsupervised clustering, supervised classification, and automated detection. A difference between both unsupervised clustering and supervised classification is use of computers and input and output reference to the sound. Unsupervised classification is processed without the need for labeled data and uses machine learning algorithms to cluster data. Supervised classification is a machine learning approach where the algorithm knows where the sound is coming from. For automated detection, we use binary classification, where in this study uses presence and absence, whether there is a gibbon or not. We use a collection of methods to measure variation within and between individuals and across populations. The training data is collected using ARUs set continuously for 24 hours after the start of dawn from the Jahoo area and are manually annotated through annotations first for training data and then for data augmentation transfer for automated detection using BirdNet transfer learning. Programming languages such as R and RStudio are used for supervised and unsupervised classification, using random forest and HDB scan algorithms respectively. The automated classifiers can be used to create visual graphs, including the ones illustrating call time and frequency, as well as territory use in respect to ARUs. One of the principal questions to ask is how can we tell females apart using supervised classification? For the supervised classification using random forest algorithm, we randomly divided our pooled data set into a 70-30 split 25 times and calculated a balanced accuracy due to uneven sampling size with a mean accuracy of 0.68 with a standard deviation of 0.03. The manual annotations of recordings using GPS was 724 hours, which resulted in Group 1, with 829 and Group 2, 135 high-quality calls. The plot to the right is a UMAP biplot providing 2-dimensional visualization that simultaneously displays the relationships between data points and their features in a reduced dimensional space. Each point is one call, and color is individual ID. We do not see strong evidence for clustering by individual. The automated detection at Jahoo for the middle territories has approximately 4,000 hours of recordings, with a total of 790 high-quality calls for Group 1 and 71 for Group 2. The automated detection of calls in a green array was around 3 kilometers away and had around 1,700 hours, resulting in 236 high-quality calls. The BirdNet transfer learning had approximately 4,000 hours of recordings with 790 high-quality calls for Group 1 and 71 for Group 2. The UMAP plot on the left is colored by individual ID, Group 1 and Group 2, and the calls from the other array. The plot on the right is colored by unsupervised clustering assignment. There are 3 clusters, but they do not correspond to individual ID. To wrap up, our balanced accuracy was only slightly higher than chance. We use MFCCs, or male frequency septal coefficients, as features, commonly used feature extraction approach. There are remaining questions to consider, such as if there's less individuality, feature extraction methods not capturing variation, and finally, does the presence of co-singing influence our results? Additionally, we will look at whether the presence of co-singing impacts the ability to classify and differentiate individual or group calls. Ultimately, we hope to eventually use PAM to identify a number of calling individuals in a given area. With that, I would like to close and give a special thanks to Cornell Summer Experiential Learning Grant. It would not have been possible without the opportunity to delve into research at depth. Thank you.